without further ado, I'm going to hand over to our keynote speaker, my friend and colleague, Andrei Irimia from the University of Southern California. And we were just speaking before we went live about the current situation in the vicinity of USC. So I hope everything is well and safe for you out there. So Andrei, apart from being at USC, was a visiting professor at the Center for Healthy Brain Aging at King's College London. So welcome you back to King's to give this keynote talk. And thank you again for joining us. Andrei will be speaking about neuroimaging and AI for healthy brain aging. So over to you, Andrei.
Thank you very much, Richard. And can you guys hear me okay?
Yep, perfect.
Okay, perfect. All right. Well, thank you so much for having me here today and for inviting me to give this keynote talk on neuroimaging and AI for healthy brain aging. And as Richard indicated, I'm an associate professor in the Leonard Davis School of Gerontology at the University of Southern California. And this past year, I've been a visiting senior lecturer at King's College London. So it's my pleasure to give you this brief overview of our research and in relative to our efforts to translate solutions on neuroimage analysis to the clinic. So first of all, as you heard our previous speakers, diseases that involve the aging brain pose tremendous challenges for healthcare systems. In particular, Alzheimer's disease and neurodegenerative diseases involve tremendous expenses in industrialized societies, and there is a lot of interest in utilizing neuroimaging models, electrophysiological models, and other approaches, genetics, omics, to understand risk for Alzheimer's disease and to evaluate this risk as early as possible during one's middle age in order to identify persons who could be at highest risk for disease. And neuroimaging has a key role in this task because it provides a lot of non-invasive information about the aging brain, and features of neuroimages can be used to estimate risk for disease, and the rate of brain aging is associated with disease risk. And so this information can be very valuable, but it should also be interpreted very carefully and in appropriate context. In the past 5-10 years, there's been growing integration of AI with computer vision approaches to quantify brain health and brain aging in meaningful ways. There's been a lot of research and ongoing work on estimating risk for disease from neuroimaging models and creating 3D models of the brain that can identify features that are predictive of Alzheimer's risk. And so my lab at the University of Southern California is focused on interpretable and ethical AI to estimate dementia risk and brain aging, to identify brain aging trajectories that could lead to dementia. And so our aim is to move from reactive care in individuals who are experiencing cognitive decline or cognitive impairment to proactive interventions that can be personalized to identify and estimate the risk for Alzheimer's early on, before the onset of cognitive decline, so that individuals who are at the highest risk can benefit from personalized interventions or other strategies to reduce their risk and to delay or even completely remove the risk of their conversion one day to Alzheimer's disease. And the previous speaker had a slide on brain age, which was great, and because that gave me a great transition here. So brain age reflects the biological aging in our brain that could either be in excess of or under the level expected for one's chronological age. So if we look here on the left, we have if a person who is chronologically of age 35, but biologically of age 40, that suggests a shorter health span and potentially a higher risk for neurodegenerative diseases. If that same person, however, has a biological age of 30, that suggests a longer health span. And so that indicates how a biological age, especially in our case, brain age, can suggest higher or lower risk for disease. If we take a very large population and look of persons who have the same chronological age, and we measure their biological ages, we find that these ages fall on a Gaussian curve. And so on average, the biological age in that population of individuals who are chronologically of age 38, for example, is going to be 38. So the biological age on average will be about equal in a control population to that of persons who are chronologically of age 38. But you see that about half of these individuals have biological ages that are younger, and about half have biological ages that are older. And traditionally, people have used different values, different features of MRI, of brain MRI, like cortical thickness, surface area, to do regression models that can predict the brain age of the individuals. But in more recent years, people have switched to AI to identify features automatically, potentially very complex features that might be indicative of a person's biological age. So one approach that's been very successful is using a type of architecture called convolutional neural network, which takes in the images of the brain and looks at different scales of the anatomy, of brain anatomy, across the layers and blocks of the convolutional neural network. And at each of these scales, the AI is able to identify features of the brain that might be indicative of biological age. And so a few years ago, we had a paper to do this kind of research using interpretable AI, which, as I'll show later, is useful to the clinician because it provides more confidence and explainability into the causes for which different features of the brain are being used by convolutional neural networks in different people to identify brain aging. And now I would like to show you just a very quick video that features this research that was done by the National Science Foundation. So let's see if I can view this, if I can play it. Okay, I don't think I have the ability to run the video, so I may have to skip this.
Andrei, I'm sorry, we don't hear sound.
Oh,
You may need to, in some way, share the sound. The video is playing well, but we don't hear anything.
okay. So sorry about that. I'm not sure what to do. I might have to skip this. I'm sorry about that. Um, it was a video from the National Science Foundation to illustrate this research and the study we had. So I'll just skip to my next slide. One feature of our work that we're very proud of is the ability to use an interpretability framework to provide insights to clinicians into how neural networks make their decisions about which features of the brain are best indicative of a person's brain age. And so one approach to do this is called saliency mapping, which looks at areas of an image that are most relevant to the decision that the AI is making. So one very simple example is from a neural net that aims to classify the animals in a picture according to whether they are cats or dogs. And so with saliency mapping, the researcher is able to identify which parts of an image are most relevant to the decision to the binary decision as to whether the animal being portrayed is a cat or a dog. And as you can see, there are several methods to do that, and most of them agree in identifying features of the animal's snout or head that are most relevant to the classification. So we've implemented a similar approach to our work on estimating brain age. And in this case, we identified parts of the brain that are most relevant to the estimation of biological age. So we benchmarked 7 saliency methods that have been proposed in the literature for anatomic interpretability. And we found that all of them identified known brain aging features such as deepening of the sulci, enlargement of the lateral ventricles, brain atrophy in general. And we found that among the 7 we had looked at, a method called integrated gradients, which is shown in this column, probably had the best, most robust performance against a series of quantitative metrics. We also found that some methods actually mislocalize saliency outside the brain, and so that's probably not very useful for a clinician. And so we want to identify methods like that that could potentially result in improper categorization or characterization of brain aging. And so this interpretability approach that our approach is equipped with supports the deployment of ethical AI and improves regulatory confidence in our work. Another important feature of this is that our approaches for brain aging mapping and for brain age mapping is able to identify patient-specific features of aging. And because it can do so, it can also enable group analyses of differences in brain aging across different types of cohorts. So for example, here we're looking at sex differences in brain aging, which are very important for the study of neurodegenerative disease, where there is a considerable difference in risk, for example, in Alzheimer's disease risk between males and females. And so when we map these saliency differences in brain aging between males and females, we find that males have higher saliency in frontal areas, in temporal regions, and that could be related to motor decline that's been documented in males to be more severe with aging. Females, on the other hand, have more saliency in certain occipitotemporal areas and insular cortices. And if we look at these maps of differences between males and females in anatomical aging, and we map those onto the known patterns of cognitive aging that have been documented in the two sexes, we actually find that the anatomical differences in aging that our model estimates map onto patterns of sex differences in cognitive aging. So there is certainly a lot of potential in using this kind of maps to identify group or subject-wise differences and heterogeneity in aging patterns. Another method that we have introduced just recently this year in a paper in PNAS involves mapping the pace of brain aging. So brain age looks at cumulative aging over the entire lifespan. but the pace of brain aging is more responsive to contemporaneous changes in a person's rate of aging. So the pace of brain aging looks at the temporal rate of aging rather than the cumulative amount of aging that that person has experienced. And there have been measures of aging from DNA methylation from epigenetics data, but those are based on whole blood cell measures, and these whole blood cells do not cross the blood-brain barrier, and so they have weak correlations with measures of DNA methylation of cells in the brain. And so using MRI, we were able to look at mapping the pace of brain aging. And we found that actually our longitudinal AI model that estimates the pace of brain aging is actually about 7 to 10 times more accurate than traditional brain age models that could be used for the estimation of the rate of aging. And we found in our PNAS paper that faster pace of brain aging is associated with worse adverse cognitive change. And so the method we developed is actually mapping changes in cognitive performance across chronological age, and so that could be used in an additional biomarker for estimating a person's risk for Alzheimer's. And finally, we find that region-specific features of the brain that are indicative of the pace of brain aging reveals differences in risk patterns according to age, sex, and cognitive impairment status. And so that's another important insight that can be used to estimate risk for disease. The last exciting approach that I'd like to show you involves predicting what a person's brain is going to look like in the future. So we can look at a person's baseline MRI scan, and using a generative AI model that synthesizes images of the brain for a person into the future, we can actually generate future MRIs from the baseline scan using an architecture that combines a diffusion U-net with a control net. And so this measure actually captures regional atrophy and predicts what a person's future regional atrophy is going to look like over time. And the predictions actually match the person's follow-up MRI with an accuracy of about 93% currently, and we're working on a better model that's likely going to improve these estimates. And so these measures for estimating future risk are probably going to help early risk identification. And when these measures are integrated with clinical, volumetric, and demographic, as well as genetic data, we're actually finding that our dementia risk models that integrate these measures are able to predict conversion to cognitive impairment in persons who are cognitively normal with about 90% accuracy in the data sets we have studied. And because of the saliency and interpretability approach, we're able to find regions of the brain that are indicative of a person's future risk of conversion. And so these predictions are reliable up to about 5 years before the onset of symptoms in cognitively normal adults. And this technology supports stratified care and hopefully personalized prevention strategies. And so interpretable AI, in conclusion, is essential for trustworthy brain health technologies. There are known risk factors for Alzheimer's, but these risk factors alone are not sufficiently useful to do personalized prediction. With tools like ours, we can help to screen, monitor, and tailor care of individuals who are now cognitively normal, who may one day convert to AD across their aging trajectories. And our models are importantly scalable to national biobanks and clinics and could be applied in the field. And so our impetus is to build bridges to identify novel solutions and push these forward this technology to achieve ethical and actionable AI for healthy brain aging. Thank you so much for your attention, and I'll be happy to take any questions.
Andrei, wonderful talk. Thank you so much for a great overview of your research. I know we've spoken a lot about this already, but really puts the AI into the context of the MRI research that you're doing and also the pace of aging,
Thank you.
biological aging. So thank you so much for that.